Faisal. And so today I'll be giving an update from what we've been doing in foundation models in general and what we've done since I spoke here last year. I'll talk about foundation models today, and then tomorrow I have another talk where I'll talk more about the agents that we're developing using those foundation models. Here are my disclosures. So my lab predominantly works on computational pathology and trying to analyze pathology images, glass slides. Once they're digitized, they look something like this. They're hierarchical, they harbor information at many different scales. They look more like satellite images. They're an interesting aspect of this data modality is that it's a 150-year-old diagnostic modality that's only recently been started to be digitized. So all the modalities that were discovered and came about later, like genomics, transcriptomics, single cell, and so forth, were inherently digital, but pathology was sort of left behind. And because of the I think something is
what?
It's not sharing my screen anymore. Okay, now it is.
Oh, okay.
Okay, so it's a very old modality. Most of the discovery is still done. Only computational analysis is still quite rare, but it's changing. So we do a lot of this analysis, but also try to combine other kinds of data to answer some of the very specific questions we want to answer around prognostics and the response prediction. We also do really more recently a lot of work in drug discovery and development using pathology images. So pathology really touches different spectrums of biomedical research and human diagnostics. So human diagnostics is just a part of it, but looking at these glass slides, we also analyze toxicology in analyzing which compounds are fit for moving on to different kinds of trials. So this has also been a phase of interest to us. So the most common framework, when you look at this large image from a machine learning point of view, what comes to mind is that you can use multiple instance learning because you can just a very large image, you chop it into smaller patches, you lose some context, but you can still make reasonably good predictions whether you pose it as a classification problem or a ranking problem. And just like every field, the most important component here is a feature extractor, which is beginning to be in the beginning we're using a lot of resonance trained on ImageNet, now replacing with self-supervised models. So we developed a few foundation models last year that I spoke of. I gave the talk here last year. So one of them called ONI is based on the DynaV2 framework, and we use large amounts of data from our hospital to train the model. That one was a vision language model that was based on a COCA framework. Both these models were made publicly available and have been extensively used. They've been downloaded over 1.4 million times over Hugging Face. So that's largely because pathology touches so many different disciplines, all kinds of biomedical research and diagnostics, but also forensics and other plant pathology and other things that you don't think about. People have used it for all kinds of different applications, and it's been applied to over 600 different studies since we made the models available just about a year ago. So we used 100,000 whole slide images to train this model initially. There's a newer version of this model that we put out a couple of months ago, which used about 350,000 cases. But the generic paradigm has been that we focus on diversity. So I think the DynaV2 article from that showed that the diversity of data is much more important than just the quantity of data. So if we were to use all the slides that were available to us in our department, they would be heavily biased towards certain data types and diagnoses. So instead, we're going to where we look at what are the most diverse cases, go and collect those cases, and then scan them and use them for training. It was trained using the DynaV2 framework and applied to all kinds of different downstream tasks, looking at classification, segmentation, both at the level of the slide as well as at the revision level, and looking at few-shot classification. And then in parallel, we did some work in image language, just contrasting with language, just like all kinds of other computer vision fields. We used a lot of data that's available in the Cognitive and Access database to do this limited to human pathology, and by doing this form of contrasting, hoping to improve the image representations. And we showed that was possible. So this is from March last year. Since then, we've really focused on trying to see how we can expand this to slide level. So CVPR25, we should try to just expand this by using very simple techniques, mixture of experts, scanning, clustering, looking at what are the most distinctive features, and creating a slide level representation, which can then be applied to lots of downstream tasks. But this is very, very simplistic. It has some really advantages, but otherwise very simplistic. We more recently expanded this into all Titan, so this is a preprint, and the article will be coming out in the next couple of weeks. We have the model already available, but it essentially has three-stage training. The first stage is based off of iBot, self-supervised training, followed by two more stages. So the first one is using a generative model to explain the morphologic regions within the image. And then the third stage is by using the pathology report. So you contrast against the pathology report and against the morphologic description. We also use the train short, test long paradigm that's commonly used in LLMs to train on a smaller region, like 8K by 8K region, and then test on a larger region to effectively enable this. And as our models are becoming more capable, our benchmarks are also becoming more difficult. So we're looking at rare cancers, solving over 100 classification problems directly. So these plots here show what the scaling laws effectively look like and how it compares with other slide level models that are available. So this model has been out there for a couple of months now, and people have used it for all different applications. Interesting ones include looking at few shot classification, because that would enable looking at clinical trials and seeing how few samples can you use to predict, for example, which next patient is going to respond to treatment, but also looking at image retrieval and finding similar cases. So if you have a whole slide image and a single presentation for the whole slide image, you can look at what are the similar cases in a very large database. This is how a lot of pathologists typically also analyze these cases, by saying that, well, this is an extremely rare case, and it resembles a case I looked at it 20 years ago, and then you pull that and see what are the similarities. This is a more computational way of doing that and trying to find similar morphological features across large cohorts. So we've been effectively doing this anyways to discover new features as well. So how discovery happens for pathology images today, so because most hospitals are still not digital, so that people are looking at glass slides under a microscope. So they will look at lots of glass slides under a microscope, find a common morphologic feature, correlate it out, and we can do that computationally using slide level representations. So in the previous study, we showed that we're contrasting the text of a generated or what exists in the pathology report. The reason we needed to generate text, by the way, is because the pathology reports are often very sparse. They don't have really detailed, dense morphological descriptions, and any description there might also have a very deep relationship with the image itself, because it's often written at the level of the case and not at the level of the individual image. So we're contrasting with text, which is subjective, but there's lots of other data that's available in pathology that can be more objective, like immunohistochemistry, understanding special stains that exist, as well as transcriptomic genomic information that exists. So that's what we started to do. So in ECCB last year, we contrasted with special stains and INCs, contrasted HCs with ER2 stains to try to see if we can improve representation, and that was possible. This is a much more objective second view in comparison to looking at text. If that's possible, then the obvious next step is sort of to look at transcriptomics. So in this toy example, this is also from CVPR last year, where we tried to contrast between improvement in two-shot learning performance when we did that. So this was the early indication that this is the area we should scale, like contrasting the image with modalities that are sort of more objective. So that's what we did in this study. We tried to use all the molecular data that was available to us at the Brigham and GH, which includes transcriptomic data as well as all the molecular data. And by contrasting between image and molecular data, the hope was to improve the image representation. So that's what we showed, and we start to see that it could really improve representation for some of these very difficult tasks, in particular related to treatment response predictions. So there's an example for tasks that are typically just difficult and challenging, but when you have this kind of a model that you're contrasting with molecular representations, you can improve image representation substantially. And for treatment response is a difficult task to begin with, because we often don't have pre-treatment biopsies available to us, and any data set available is typically very small unless it's very carefully curated. It's changing, but this is the current state today. So then we use these models into a generative model, and I spoke about that last year. So we developed PathChat, which is a generative model that with the hope of catering to all of human pathology. So I'm going to give an update on what I've been doing in terms of training PathChat 2. So in this case, we really expanded on the amount of data that was available to us. Lots of manual data collection, going up to 5 million or so instructions, really trying to cater to every known human disease entity. And the focus has really been to have this done at every level of abstraction, so that such that the chatbot or the multimodal large language model can answer questions at any level of abstraction, whether it's at the level of the region, at the level of the entire slide, or at the level of the entire case. So it's just a comparison with the current model that we have in comparison to a lot of the other models that exist now. So even PathChat 1 does better than most of the frontier models that are trying to be more generalist. And we also brought in some agentic capabilities where we were able to look at the entire slide by interpreting the morphologic regions within the slide, and then effectively trying to scale them such that we would eventually generate a report. This is a demo for the latest version. So what we've done is effectively looked at the entire pathology workflow and tried to see where can PathChat effectively help. So one thing you can do really well is triage out all of the negative slides. So any pathology case would have lots of positive slides, lots of negative slides. This is one large high capacity model looking through the entire slide. Can we triage out all of the negative slides and push forward no more suspicious slides? And this works on pretty universally across infectious, inflammatory, and neoplastic disease. So it's just not limited to cancer. Something you effectively need if you want to have a real triage tool. Then the other capability is that because it's a generative model, you can look at a case and say which additional tests need to be ordered to diagnose a case, and it will order those tests on its own. Sorry, skipping through the video in the interest of time. And then the more sort of multimodal large language model chatbot capability, you can add any level of abstraction, whether it's at the level of the region, level of the slide, or at the level of the entire case as well. And then once that's done, you can also generate a pathology report. So so it's essentially channeling PathChat to give much more objective responses, which can also be evaluated in a holistic manner. So this is where we are currently, and we're trying to deploy this effectively at MGH. Another direction that we have been working on since last year is trying to develop these models for more fundamental biomedical discovery related applications like spatial proteomics, as well as single cell and spatial transcriptomics. But on the spatial proteomic, and we just announced a new foundation model, we're calling it Chronos, which was just announced like a week ago. We put out the model and the preprint, but it's a really team effort from multiple institutions trying to get lots of data pulled together, and then coming up with an architecture that makes it platform agnostic as well as marker agnostic, and then trying to solve a host of different downstream tasks from phenotyping, looking at artifact detection and classification, unsupervised tissue phenotyping, and then importantly looking at query and search, trying to find similar regions, which really is the basis of how this data is today analyzed today, more looking at how cells are effectively interacting and how protein is expressed, and finding those common features across. And then finally also looking at patient stratification and biomarkers. So we have some of these examples, and we're already in the process of substantially expanding this. So we've been grateful for a very large community within the Boston area, and also internationally that has supplied data for this very basic science sort of a project. We often consider patient data to be more difficult, but so it's been even more difficult to bring together so many researchers and convince them that creating a foundation model like this would be beneficial. So I do want to touch upon a little bit about what we're working on next, and we'll have more to say about this in the coming months, but I do want to share a little bit. So on a model that we're calling Apollo, and we have already results for this. We're trying to use data from basically everything we have at our hospitals. You have data corresponding to 10.1 million patients. I guess at this point, around 10.2 million patients from our 12 hospitals. It's really a unique setting because the Harvard hospitals get some of the most unique, challenging cases. We have very rare disease, a lot of diversity that other hospitals don't see. around the world, and use all this data into a paradigm where we're building patient level representation. So lots of model representations that get get fed into a patient. It's also effectively temporally aligned, such that the embedding gets continuously updated. So we have started working with this model now, and we're looking at lots of interesting aspects, early detection, prediction, discovery from it, and also working with some pharma partners to see if we can do clinical trial matching from this model, and have a lot to say about this in the weeks to come. So I'll stop here in the interest of time, but I'd like to thank all the PhD students, postdocs that worked on projects, as well as all the funding that we received in this work. Thank you.